So how do we use oligo pools to generate and build DNA libraries, and in particular, antibody libraries? So if you think of an antibody variable domain, either a heavy chain or a light chain domain, is essentially a collection of three different loops that are pieced together. You could think of basically synthesizing an oligo pool that encodes for diversity in each of those different loops, and then basically seamlessly PCR them together to create a hypervariable domain. I know this is subtle, but the reason why this is in my mind kind of game changing in the antibody engineering and discovery space is that in the past, when people have made synthetic libraries, they typically use what's called a degenerate oligo, which is an oligo that has mixtures of nucleotides or mixtures of trinucleotides, and you basically try to use that oligo to mimic the diversity that's seen in the natural human repertoire as you build an antibody phage split library. In this case, I don't need to do that. I can actually make explicit sequences in a pool and then just shuffle them in different contexts within the context of a single human germline framework. So this allows me to make libraries that are much more precise and also that exactly match the natural human antibody repertoire if I choose to. In addition, I can also remove sequences up front that might lead to developability risks. So isomerization sites, cleavage sites, deamination, like glycosylation sites, for example. And then finally, if I choose to, I can also code for specific motifs in our CDR sequences as well. And that's actually really important for our GPCR libraries, which I'm not going to talk about today, but if you're interested, please again follow up with me. So again, just to reiterate the power of the Twist platform to make libraries, again, we use this silicon-based DNA synthesis platform to make huge pools of oligos, and we can use those pools of oligos to make really precise antibody DNA libraries, in particular antibody phages for libraries, where we have really strict codon usage control, and we can control the combinatorial diversity of the combinations of those different oligo pools in the construction of the library. We have tight control over the amino acid distribution in each position. And because we're just making pools of individual oligos, we can actually, it's very easy to modify and have different lengths of CDRs, which again, using the traditional ways of using degenerate oligos, it's actually really difficult to do and you require multiple degenerate oligos. Of course, we can avoid restriction sites and unwanted motifs, potentially use multiple germline frameworks. And finally, last but not least, you can also validate the library at the end using next generation sequencing to make sure that the final library matches the design that we intended. So what is the Twist Biopharma vertical within Twist Bioscience? We're basically an antibody discovery and optimization group that utilizes all the fantastic DNA products within Twist to help pharma and biotech discover as well as optimize antibodies. And we basically have two kind of flavors of antibody libraries. The first one are just kind of general use libraries that are either based off of Fab, single chain FE, or VHH scaffolds. I'm going to talk today about the VHH libraries and how we use them to discover novel antibodies. But we also have a whole series of libraries that can be used for difficult to drug targets. I'm not going to talk about today. Our primary difficult to drug class of targets are our GPCR libraries, which we actually have two. And we're continuing to innovate in this space to create libraries against ion channels and other things like carbohydrates as well. Thirdly, we have a whole platform for doing antibody optimization. I will also talk about this today as it relates to doing affinity ranking and binning. So again, a fantastic system that we have in-house for doing affinity maturation and humanization of antibodies. And then finally, I'll also mention our new alpha product, which is our ability to do high throughput antibody production. It's part of every project we do within the Twist Biopharma vertical, but it's also a product that we're thinking about rolling out to the greater Twist customer base. And so please stay tuned for that update. So again, thinking of ways that we are differentiated from other antibody discovery companies out there, one way that I think that we're very special is that oftentimes in the phage display arena, you're limited by the overall diversity of your library. For a phage library, that's typically around 10 billion. So if you want to increase the breadth of your diversity, rather than just building a bigger and bigger library, one way is actually to build more libraries. So my solution to this is because Twist can build high quality libraries so quickly, why not have at one point in the near future, a whole plate of libraries where we have basically 100 libraries of 10 to the 10th. So overall, we would have a total diversity of roughly 10 to the 12th. So that gives us a huge breadth of different scaffolds, different diversities in CDR loops that allows us to, at the end of the day, be successful against any target that comes our way. And so I've coined the term a library of libraries that Twist Biopharma is offering.